Okay, thank you, Andy, and thanks to all the organizers, especially you and Song, for arranging such an excellent meeting. I'm going to talk today about three topics related to the work done in my lab on the Neanderthal Genome Project. This is, of course, a very large collaborative project headed by Svante Pääbo and David Reich, and involves many other people. I will focus on things that were done primarily in my lab, one published and two unpublished. Recently there was this paper of a Neanderthal fossil found in the Denisova cave in the Altai Mountains. Here, it's the same cave from which the Denisova fossil was found, but at a slightly lower stratum. This turned out to be very, very high-quality fossil, allowed sequencing to a depth of 52 or 53x, and nearly a complete coverage of the single copy DNA. There are many interesting things about this, the genome sequence from this fossil. I will focus on the one that we were most involved in. When Heng Li and David were doing their analysis using PSMC to try to infer the demographic history of the population from which this fossil came, they noticed that there were appeared to be long runs of homozygosity, suggesting that this individual was highly inbred. And so Flora J, a postdoc in my lab, and I took on the project of trying to understand in more detail exactly what the inbreeding was in the ancestry of this individual. And there were a number of technical issues that Flora had to resolve. One of them was to decide exactly how you call a run of homozygosity. If you require perfect homozygosity, then you will, sequencing error, alignment error, and other issues will produce what appear to be heterozygous sites in the middle of long runs, breaking up what seems obviously a long run into several shorter runs. And so quite a bit of simulation effort on Flora's part went into deciding exactly what criterion to use to call a run of homozygosity. Once she arrived at that criterion, and also you have to deal with missing data and other issues, then indeed we found that there were a number of very long runs of homozygosity. Here are two chromosomes. We're showing only runs of homozygosity longer than 2.5 centimorgans, where we just estimated the distance in centimorgans by converting megabases into centimorgans. We did not put this on a real recombination map. And you can see, for example, two things here. On chromosome 8, there were many of these shorter runs of homozygosity compared to what we saw in the Denisovan genome and what we saw in a high-coverage, high-quality Papuan genome that David's group had sequenced. And this was typical of what we saw in the other chromosomes. In addition, there were many very long runs. This stretches more than 40 centimorgans. And so we concluded two things from preliminary analysis. One is that there's obviously a lot of inbreeding in this individual, but there may well have been two kinds of inbreeding. There was inbreeding due to a very recent common ancestor, incest in the Ice Age. But there was also inbreeding possibly due to population structure or more, we don't know the cause, of course, but there may have been what we call background inbreeding. So in addition to having an immediate common ancestor or two, there was more distant common ancestors. And so to sort this out, the first thing we did was look at the distribution of the lengths of homozygosity fragments. We call this homozygosity by descent. And compared this to the Denisovan and the Altai genome. Notice the scale is very different. So in the Altai genome, in the Denisova genome, there's only one run of homozygosity greater than 10 centimorgans. Here there were many more, 16 or 17. In addition, there were more shorter runs. The graph doesn't show that as clearly. So it was obviously inbred. About 13% of the genome was in runs of homozygosity greater than 5 centimorgans. And so Flora did then we tested a number of family structures, all the possible family structures that could lead to the level of inbreeding we thought was appropriate for this individual. And to separate the recent from the background inbreeding, we focused first on tracts that were longer than 10 centimorgans, because those could only be a product of recent inbreeding, or at least the number we saw could only be a product of recent inbreeding. And she simulated these various family structures. All of these have inbreeding coefficients of 1/8, and the asterisk means also grandmother, grandson, or aunt and nephew. These are avunculate marriages here. We considered full siblings, and then half uncle and niece, and first cousins. These have an inbreeding coefficient of 1/16, and these have inbreeding coefficient or relatedness of 1/16 and 1/2. And so with these simulations, she could match the data, the number of runs of homozygosity, and the total coverage of the runs of homozygosity for these long runs with any of these 4 families, meaning that it's most likely that the inbreeding coefficient of this individual was 1/8. But we could not distinguish between any of these family structures, even though we are quite aware they involve different numbers of common ancestors and different numbers of meioses separating, going down the different pathways. So it's not that we didn't try, it's just that we tried. The recombination in a few generations is so stochastic that you don't get a clear signal that allows you to distinguish this, at least using the methods that we have. Remember, we don't have allele frequencies in the population to use that information. We think it's very unlikely that the parents were full siblings, and it's also very unlikely they were first cousins, but we can't absolutely exclude these. So, but undoubtedly there was very recent inbreeding. And that's what this conclusion section says with some qualifications. We then looked at the X chromosome and found at least one run of homozygosity greater than 10 centimorgans. I think it was 16 centimorgans, meaning that the X chromosome itself was inbred. And if you remember your basic genetics, that allows you to exclude certain families that could lead to an inbreeding coefficient of 1/8. The most obvious one is you can exclude this family, where these parents are half siblings with a father in common. The reason is that the X is not transmitted down both paths. So you could not have an inbreeding coefficient on the X as the same as the autosomes. If the parents were full siblings, half siblings, then it's the mother, not the father, in common. And there are some other more obscure ones, but this was the most important. So in addition to the recent inbreeding, we, as I say, we suspected that there was background inbreeding. And to look at the background inbreeding, we analyzed the coverage for tracts in the range 2.5 to 10 centimorgans. Remember, the recent inbreeding was based on the 10 centimorgan and over, so we're not reusing information. And what Flora found is that once you subtract the expected inbreeding, the expected number of tracts in this range attributable to recent inbreeding, there were way too many to be consistent with the assumption that there was no other inbreeding. So if there was no other inbreeding, these lines would be down here. But under any of the family structures, the expected number once we subtract the expected number of tracts in this range attributable to recent inbreeding, we have too many left over. So we concluded from this analysis that not only was this individual highly inbred because of her recent family structure, but there was background inbreeding of some kind. Now that background inbreeding could either be that the parents were themselves somewhat inbred. So in the family structure, in this family structure, for example, these two males were themselves close relatives and had a common ancestor up here, or the mother was somewhat inbred. That is, you had not only a recent common ancestor, but more distant common ancestors. So that's one possibility. The other is that there was simply a very small population, so everybody was somewhat inbred. And we couldn't distinguish between those. Flora had a very difficult time finding a scenario of successive bottlenecks and constraints on the population size, which gave us the level of background inbreeding that we observed in this individual. But she couldn't exclude the possibility that there was some purely demographic cause for it. So there was background inbreeding. We don't know exactly why. To answer a question that I was asked a number of times, is this why Neanderthals went extinct? The answer is, I don't think so. It's more a sign that they were a small population, which the PSMC analysis also showed, and had been a small population for a long time. But I think that was a symptom of the problems that they faced from climate or competition or just plain bad luck, rather than the actual cause of their demise. Once you say that, reporters lose interest in a hurry in talking to you. So here's the background inbreeding. That's our conclusion. And that's all I'm going to say about the inbreeding story. Second part of what I'll talk about is new work done by Fernando Racimo, who's here someplace, I hope. Yes, there he is. And this is, we wanted to use the information from the Altai genome, the Denisova genome, to look for selective sweeps in modern humans. And this is something that a number of people have been concerned with. We wanted to see if there were methods for analyzing the data which took account of the information from the Neanderthal genome in addition to the more conventional methods of analyzing for selective sweeps of the kind done by a number of people here, including Chuck and Molly and Wolfgang and Rasmus and others. So there's a lot of work on selective sweeps. What we're trying to do is add information from the Neanderthal. So the idea is we had, of course, modern humans. We focused on sites that were fixed derived in modern humans and fixed ancestral in Neanderthals and Denisovans, because they seemed like good candidates for, and also sites that were in interesting regions, in coding regions or regulatory regions or splice site regions or so on. And if they were fixed in all modern humans, they had to have been fixed before the separation of modern humans into different geographic groups. And we wanted to be able to detect hard selective sweeps that arose very early in the history of modern humans, so they could go to fixation by this time, or possibly even were fixed alleles that were in low frequency in the common ancestor and then increased in modern humans. And the currently available methods, the method used by Hernandez et al. And other studies of selective sweeps in humans have, as they say, they have the range of about 10,000 generations. If a sweep is finished more than 10,000 generations in the past, I believe there's no power using currently available methods to detect those sweeps. So I will call things that, sweeps that finished more than 10,000 generations in the past, ancient sweeps. And so what we did was use some summary statistics which are designed to target these ancient sweeps. One is a measure of heterozygous, well, let me go back. The idea to detect an ancient sweep, one thing a selective sweep does is derive the ancestral haplotype to a high frequency. We all know that. That comes from Hagen Maynard Smith. But then after it's fixed, you have very close to the selected site, you have you have the single haplotype fixed. And then you can start to accumulate mutations, new mutations that arose after the sweep was completed. It doesn't always happen, but if it does happen, what you will produce is a very distorted frequency spectrum of haplotypes. And so we focused on sets of segregating sites, sets of 4 segregating sites near these candidate sites that we were concerned with. So if those sites had been driven to, if there was a high frequency haplotype for those sites, then evidence of a past selective sweep might be in the frequency of the most common haplotype, something Dick Hudson looked at many years ago, or the presence of a lot of rare alleles. So basically a distortion of the frequency spectrum as seen from the perspective of the haplotype. And so a measure like this, this isn't pi, this is the heterozygosity of the 4 sites that we are considering, and we used a sliding window. But this would be low if those sites were in low frequency. The segregating sites had a low frequency allele. And then also, these are of course highly correlated, the haplotype frequency distribution would be skewed because you would have one common haplotype and a lot of rare haplotypes. And then finally, we tried to include information from the Neanderthal genome, Denisova genome, in the following way. One thing a selective sweep can do, it can take a haplotype that's initially rare and drive it to high frequency. I mean, most likely the haplotype the advantageous allele is on will be one of the common haplotypes. You just get the luck of the draw. But there is the capacity to take a haplotype that's initially rare and drive it to high frequency and make it the most common haplotype. If that happens, then that haplotype was initially rare. It was probably rare. It will probably be rare in the Neanderthal genome as well. So you would have the most common haplotype not present in the Neanderthal genome with a slightly higher frequency if there were a past selective sweep, an ancient sweep. Now, of course, we don't have phase relationships in the Neanderthal genome, but we could ask, we did have phase data. We used the 1,000 genomes phased data. We could ask, is the haplotype that's in high frequency for 4 base pairs consistent? Could it possibly be one of the Neanderthal haplotypes? So if it could possibly be one of the 4 with any permutation of the Neanderthal, then you'd say, yes, it's consistent. If it's inconsistent, then you'd say it's inconsistent, if there's no way for it to be one of the Neanderthal haplotypes. So we had an index of consistency, which was either 1 or 0. We didn't try to say how consistent or how inconsistent. And then Fernando did a large number of simulations, just looking at the properties of these 4 test statistics as a function of the time since the selective sweep was finished. So this is 13,000, 9,000, 5,000, 1,000 generations. This is really the ancient sweep column. And looking at the properties of the 4 test statistics, this is the majority haplotype, this is the index of evenness, this is the heterozygosity, and this is this index of consistency. And you can see that, for example, the evenness doesn't show much of a signal for an ancient sweep. The majority haplotype doesn't. The heterozygosity, this computed heterozygosity, shows a little signal. It's there. It's not just a printer error. And the inconsistency surprisingly shows a substantial signal, even for a sweep that finished 13,000 generations in the past. So this gave us some hope that even using these test statistics without doing anything really hard, could provide some information about selective sweeps.
So,
Question?
This normalized by the mean, not the standard deviation?
Fernando?
Sorry?
What, is it normalized by the mean?
Yeah,
not-
it's normalized, it's relative to an outside neutral region and then divided by the standard deviation.
Oh, okay, so also.
Okay, so the next thing we did was actually...
Is
I'm
there
sorry.
some like intuitive explanation for why it's the signal's lasting there? I mean, are the recombination rates so low within?
Well,
that?
these are four adjacent polymorphic sites
So how close
so
is adjacent? It looked like they were like
within
a
10
centimorph,
k
didn't it?
within 10 kb. aren't they?
Okay. Okay.
So the first thing we asked is there any power to reject neutrality just using one of these test statistics on its own? And these were scaled in different ways. H prime and H double prime are two different scalings. One, they're scaled partly using the differentiation of those four from the chimp genome, and partly using the distribution in the population, the standard deviation in the sample for regions not near the selected site. And we found if selection was strong and so S is 0.01 and the sweep finished 13,000 generations in the past, then there was some power even using just one of the indices. So H double prime is a scaled version of this compatibility index. This is a slightly different scaled version, not as good. There's some power, I mean 25% power. It's not great, I mean it's not what we had certainly hoped for, but it's better than nothing, or at least we thought it was better than nothing. With and it does have considerable power for sweeps that finished in this 5,000 to 10,000 range, which is already hard to detect. So it wasn't as I say, it wasn't as good as we had hoped for, but it's better than it could have been. With weaker selection, selective sweep of 0.01, a selection coefficient of 0.01, then we really don't have any power to detect ancient sweeps. My argument is that these are the sweeps that we're really interested in, the ones that were driven by whatever new conditions humans experienced early after the separation from Neanderthals. So then Fernando looked at, did an ABC analysis, approximate Bayesian computation analysis, using all four indices together, the H double prime version, and simulated various sets of, under different sets of parameter values, and grouped the outcomes by the Bayes factor that separated neutrality from selection. And so with using a Bayes factor of 5 and a selection coefficient of 0.1, we could reject neutrality about 40% of the time if there really had been a selective sweep of that type. So we were fairly pleased with this, and for weaker selective sweeps, it doesn't. We don't have nearly the same power in this range, although we have considerable power in this range. And when you simulate neutrality very nicely, we don't get much of a signal of selection. That's also convenient. So then Fernando applied this to the data from the Neanderthal genome paper, the Altai paper that I showed you, with this number of sites. These are the sites of each category that were fixed in modern humans and ancestral in Neanderthal and Denisovan. Unfortunately for us, we didn't find a strong signal of selective sweeps in any of these categories. There were some individual sites that had significant Bayes factors, but like the Hernandez et al. Paper, there was no difference in synonymous and non-synonymous substitutions in coding regions, making us suspect that, either there's a lot more selection on synonymous sites than previously supposed, or a lot less selection of this type on non-synonymous sites. So I would agree with their conclusion that hard sweeps are very infrequent in the history of modern humans. The one exception is that for splice sites, there is a consistent signal of ancient selective sweeps. that we do reject neutrality for them more than for other types of changes. Whether that means something, we don't know. So with this analysis, Fernando, then these are the sites that had a Bayes factor greater than 5 in favor of selection, and also a good fit to a model of selection. Some of 3 of 4 of them are non-synonymous, several splice sites, and some other sites in the regulatory region. So, and again, what this means, we're not completely sure. So that's the second part of my talk. The third part is work that's not published and not even completely finished. It's being done with Melinda Yang, who is also here someplace, back there, yes, trying to go as low in the seat as possible. We call this projection analysis, and it didn't start from the Neanderthal Genome Project. It started from another project that we're collaborating with David and Johannes Kraus on the genome sequence of a fossil, 8,000-year-old modern human fossil from the Lashbour cave. The manuscript describing that work was just put on the archive a few months ago. This work didn't go into that paper, but it was motivated by it. Because the question is, suppose you have a genome and you know nothing about it. You just get a sequence from the sky. And that's what the Lashbour genome was. We didn't know what it was. It was just something. It was a genome. But you do have populations that you know a lot about. You have 1,000 genomes data. And so you would like to know, in some sense, the relationship between the single genome, and I'll try to be consistent and call that the test genome, and one or more populations, and I'll describe only one population at a time, and say, well, we have a reference population, and you can think the 1,000 genomes, European, African, Asian population, or some other population that you happen to have sequenced. Drosophila melanogaster or Drosophila pseudoobscura or some reference population. And you would like to just see the relationship. And I view this work, this method, more as exploratory data analysis than it's certainly not a method of estimation or hypothesis testing, at least yet, and it may never be. But it leads to some interesting ways to think about data, and so I will describe what we've done in that regard. So you take every polymorphic site in your reference population. So you've computed your frequency spectrum, you know the allele frequency at every reference site, every site in the reference population for every segregating site. Now you go to the test genome and you say, what is the state of the test genome? If it's homozygous ancestral, then you give a weight 0. W is the weight you assign to that in the test genome. If it is heterozygous, then you give a weight 1 over 2x, where x is the frequency of the derived allele in the reference population. And if it is homozygous derived, you give it a weight 1 over x. It seems a very curious thing to do. It will soon be obvious why I'm doing this. And then you collect all the sites in the test genome which have the same frequency of x in the reference population, and you compute the average weight. And I will call this average weight, W bar of x, the projection of the test genome onto that reference population. If you have a different reference population, you get a different projection. If you have a different test genome in the same reference population, you get, of course, a different projection. Now, the reason maybe it's obvious to all of you already why we did it this way. If the test genome is a random sample from the reference population, the projection is 1, independently of x. And this is one example of what I call Felson Steins Law. Everything that's true is obvious. And this comes from years of explaining things to Joe. I think I have some new nice result, and I explain it. He listens very patiently, and he says, "Oh, well, that's obvious. It's because of this." So that's Felson Steins Law. So it is, I think it will be obvious that the projection of a population onto itself is one. If the relationship between the test population, that is the population where the test genome came from, and the reference population is represented by this tree where there was a clean separation tau generations in the past, then work that I had done years ago with Hua Chen just modifying the algebra a little bit, shows that the projection decreases exponentially with tau over 2n. This is if all the populations are of constant size, but is still independent of x. So there's a line and it just settles down at a rate that depends on the time of separation. That's slightly less obvious, but it doesn't take long to agree with that. Okay, now we start to ask about more interesting demographic events. Suppose there was a bottleneck in population size in one of these populations. How will that affect the projection? Well, we couldn't do any more analytic theory, although I should say at this point, what we're doing is really looking at one aspect of the joint frequency spectrum of these two populations. And and this is very close to what Nick Patterson will talk about in his talk, where he has a very sophisticated, useful analytical method of studying the joint frequency spectrum. Well, we didn't use that. We used the Ryan Guntenkunst program DADDY, not the inferential part, but we used it to solve the coupled diffusion equations. But it's consistent with results we got from doing coalescent simulations. Okay, so remember, the question is what happens with a bottleneck? If you have a bottleneck in the test population, the projection is still flat. We assume here all the parameters are motivated by the Neanderthal example, so the time of separation is 60,000 years, population size is 10,000, 25 years per generation, and we assumed a bottleneck reduced the population size by a factor of 10 for a period of 30,000 years. And this is roughly consistent. I mean, it's of the same order of magnitude as the bottlenecks indicated by the PSMC analysis. So there is, I mean, this isn't meant to reproduce human history, but we're trying to give it a certain truthiness, as people used to say. If there was a bottleneck in the reference population, there's a distinctive signature of that. And if there's a bottleneck in the test and reference, of course, you get the same signature. If there's a bottleneck in the common ancestral population, you get a slightly different pattern in the projection.
Would you just remind me what X is?
X is the frequency, the SNP frequency in the reference population. Now, it's think 1,000 genomes, think European 1,000 genomes population. So you measure this, and then you query the test genome to see what the state of all the segregating sites are.
And this is that site's expected contribution to W bar?
No, it's the contribution. It's the average of W for all the segregating sites averaged over the test genome. Okay, so now we come to the question of gene flow. And the direction matters. And we calculated the effective gene flow. Here is roughly the Neanderthal admixture rate about 50,000 years ago. We also considered a 20,000 year period of admixture of one-way migration from the test to the reference. If there's admixture from the test into the reference, that is from Neanderthals into modern humans, then you get an elevation of the projection. If there is migration the other way, only from the reference into the test, there's no effect. And there is no difference. We adjusted the parameters so that there is no difference between a prolonged period of gene flow and a single pulse of gene flow at a specified rate. I'd actually thought there might be a way to distinguish the two, but obviously there isn't. Okay, and I wanted to ask specifically about this situation here where you have a bottleneck in the reference population. You have no bottleneck in the test population, and you have recent admixture into the reference population. By recent, I mean 10,000 years ago. So just to these, if there's no bottleneck, this is gene flow 10,000 years ago. If there's no bottleneck, there's only a very slight effect on the projection. If there's no migration, then this is a curve I showed you before. But the combination of a bottleneck in the reference and a little bit of recent gene flow gives you a substantially elevated projection. So the last case I will consider is one where there's a reference population, a test population, and another population from which you do not have a sample, or at least you're not considering that sample. And I'm using Peter Beerley's term of ghost population to represent the thing you have not sampled, which is influencing the things you have sampled. And the ghost could admix with the test population, with the reference, or with the common ancestor. Or it could, we actually considered the case where it admixes with both the test and the reference at the same time. Now, if the ghost admixes with the test population only, it has a very slight effect on the projection. You can see that this line is the theoretical expectation, and it's slightly below it, but it's essentially flat. If the ghost population goes into the reference, admixes with the referent population, or the admixture, though, you get a depression of these alleles in frequencies that are low but not too low. And that was interesting to us because there are a number of cases where there is a claim of ghost admixture. And we wanted to look a little more at this to see if the ghost admixture into the reference, that's the one that produces the most important effect, followed by recent admixture into the test from the test into the reference. So think, for example, the ghost could be Neanderthals admixing with the ancestors of Europeans. We think that happened. And then the test could be admixture from African populations into the Europeans in the recent past. And if you allow for a bottleneck in the reference population, plus the recent admixture, plus the admixture from the ghost population, then you get a substantial effect. You don't have a depression in the low-frequency alleles created by the ghost admixture because the recent admixture and the bottleneck swamp that out. And we were curious about that. Yes, the reason we were curious about that is that we have a situation where if you only look at modern human populations, if you look at Africans as the test population and Europeans as the reference, from the point of view of those populations, there is ghost admixture from the Neanderthals. And so one of our questions was, could we see the ghost admixture without having the Neanderthal genome? And we could also test the hypothesis that Jeff Wall and others have proposed that there was ghost admixture in an African population from a population we really don't have, an archaic population we really don't have evidence of. So here's some data analysis. Now, one thing that puzzled us, all of this is using the 1,000 genome CEU population. And then the test genomes were the high coverage genomes that David Reich's group sequenced as part of the Altai and Denisova papers. These were the A panel genomes. If you sample a French using the French genome, you get the expectation. I mean, it's a European, except that you get this little dip here, and we don't know if that's. Well, we don't have the explanation for it. If you use the Han, you get a signal a little bit of admixture. The Papuan, you get a more complex signal, and all of the Africans show projections that look like this, which coincidentally, not coincidentally, we arranged it to look this way, look like this. So our conclusion is that even though this should in principle show a signal of ghost admixture, that is Neanderthal admixture into the Europeans, that is swamped by recent gene flow, recent genetic exchange between Africans and Europeans. Without assuming something like that, it's very hard to find consistency with the data.
Monty,
Yeah.
could you just go back one? What's the direction of flow here? I can't keep track.
Well, this is the data.
No, I know, but you're okay. But you're
From the
interpreting-
Africans into the Europeans.
It's from the Africans into the Europeans. That is, you need admixture both ways. No, you don't need it both ways. You need African to European admixture sometime after the European bottleneck.
Did you try North African populations? Because presumably.
No, we don't have any of those.
Okay. Well, we have some North Africans.
Well, send them along.
Send you the data, not the North Africans.
Yeah, no, please, please, yes, no. Poor Mel was sitting there saying, "Oh God." Wait, we're going here. So here's the data. Interesting, the Loschbohr, this was the motivator for all of this, has this same dip. It looks like a random European, which is why we didn't try to get this in the Loschbohr paper. The Altai and the Denisovan, remember this is the European reference, they do show a strong signal of admixture. The lower level of the Denisovan is because of the separation of Denisovan and Neanderthal. And I'm running out of time, so I won't elaborate on that. Now when you use the African, the Yoruban as the reference population, and look at the projection of Europeans, Asians, and Papuans on the African population, you do see this dip that we at least found was consistent with admixture from another source, from a population not represented by a sample. So this, I would say this is consistent with what Jeff and others have said, that there is evidence, and Nick also, I believe, has reached this conclusion, there is evidence of admixture in African populations from an archaic population, and this method isn't going to tell us much more about that archaic population. Here, this is just for completeness. The Loschbohr genome also shows that signal. And there is a signal of Denisovan and Altai admixture in Africans, which again, I think is consistent with recent admixture between Africans and Europeans, but this time it has to go from Europeans to Africans. So there has been some exchange. I think the assumption of no exchange is not consistent with our analysis. Okay, so that's all I have to say. Thank you.
Questions?
Yes, Molly.
In the ancient suite, did you look at whether there were, quote, fixed differences between Neanderthal humans in those candidate regions, in the sense that at least some of your sweeps will have post-dated or split, so you would predict that there should be a fixed difference between your sample of two Neanderthals and whatever.
Well, I think all the sites we looked at were like that. They were all fixed in modern humans and ancestral in Neanderthals and Denisovans.
In terms of the haplotype not being represented, but that doesn't preclude individual site, there not being any individual site that is actually fixed. Does it? Or am I just thinking about that wrong? So the haplotypes are inconsistent, I understand, because is it also the case that there's always a site that shows one type fixed in Neanderthals relative to
Ask Fernando, who's sitting beside you.
So the test was run always centered on a particular fixed difference. And then for the statistic, we only look at SNPs.
I missed the part where it's centered on.
Yes. Sure.
So I'm curious, right? I tend to have a knee-jerk reaction against new summary statistics because you know, there are lots of different scenarios that we could come up with.
Yeah,
that-
they're like baby pictures. You
Right,
make your own
exactly.
much better than anybody else's.
So, is there a sort of goodness of fit test or something that one could do to compare scenarios and see, or other
Yes,
additional summaries of the data?
we are doing that.
Uh-huh.
The question for us
I
is.
mean, this seems like a very,
Well,
this seems like a very logical statistic to use, and one could imagine thinking about other ways.
Right, we haven't, the question we haven't yet answered is this a better way to look for parameter estimation than simply using DADI or using ABC
I mean, it's like a sort
or
of one
something
sample
else?
daddy.
Yes, it's like a one sample DADI. I mean, I find it hard to believe that you would do better using the projection if you have the whole, you have a bunch of genomes from the test
Yeah, yeah,
population, analyzing them one at a time. I think it's much more appropriate when you have a single genome and that's all you've got.
Yeah, yeah, yeah.
Then I think that's, I mean, as I said, that's why that was the motivation, but I thought since we were in the neighborhood, we might as well look at more data. Questions? Okay.
I think Laurent had a question.
Oh,
Just wanted to ask you about this ghost input. So you're detecting some kind of ghost admixture with some kind of African ghost population, but how sure are you that this is some kind of archaic admixture, or could it be that there is within Africa some kind of substructure, and perhaps the Dinka admixture that you're seeing is maybe some coming from East Africa? So can you tell this?
well, we saw the same signal. Well, we don't really know because we don't have a population of San to test for that in the San. But to get the patterns we saw, you have to have the ghost admixtures, the ghost population diverge substantially before the European and African population. So it isn't consistent with admixture, say, between San and Yoruban. Something we've looked for that. Yes. Yeah.
Just wondering, following up on the previous question, if these methods have been ratified on some other species where you have alternative evidence?
No. I mean, I think Drosophila is the only other species right now where you'd have data of this type. No, we've tested it a lot on simulations, but we can't imagine all the possible histories that actually occur. So.